Hello everyone, my name is Alfred, a graduate student at the University of Eldoret and also a practicing biomedical research scientist. So today we are going to discuss about antigens and immunogens. This is our second upload of our ongoing immunology discussions. Hope someone finds this helpful. So under antigens and immunogens, we are going to define what is an antigen and what is an immunogen. We will also look at the features of molecules that mediate immunogenicity. We will also look at the concept of haptens and epitopes. And towards the end, we shall discuss how hapten carrier conjugate induces immune response. So an antigen can either be an organism, a molecule, or a part of a molecule that is recognized by the immune system. So antigens can also be defined as molecules that react with antibodies. And you also need to know that antigens can either be simple, complex, and some of them are protein, others carbohydrates, or synthetic in origin. Moving to immunogens, these are molecules that induce immune response, and in most cases, antigens and immunogens are used interchangeably, except for the haptens. Now, what are haptens? So a hapten is a molecule that is not immunogenic by itself, but when react with specific antibodies, they can form a new antigen capable of producing antibodies. And an antibody is a protein substance produced as a result of antigenic stimulation. So moving on to features of molecules that determine immunogenicity, we have quite a number. One, foreignness, molecular size, chemical structural complexity, the epitopes, conformation and accessibility, chemical properties, genetic constitution of the host, dosage, route and timing of antigen administration, adjuvants, and biological systems. So we are going to discuss this one by one briefly. So regarding foreignness, you need to understand that in order to be immunogenic, then the molecules must be recognized as non-self or foreign by the immune system. And foreignness implies the ability of the host to tolerate self-antigens. And tolerance to self-antigens develops by contact with them in the initial phases of the development of lymphocytes. So generally, our immune system is tolerant to molecules that are recognized as self, while it is antigenic or immunogenic to molecules that are recognized as non-self or foreign. Regarding molecular size, you need to understand that proteins larger than 10 kilodaltons are usually more immunogenic, and the most potent immunogens are proteins of higher molecular weight, especially those ones with molecular weight above 100,000 daltons. And molecules with molecular weight that are less than 10,000 daltons are weakly immunogenic, whereas for smaller molecules, for example amino acids, they are non-immunogenic. But you also need to know that substances with low molecular weight may be made antigenic by adsorbing them on the carrier particles, e.g. adsorbing them on bentonite, kaolin, and other inert particles. Moving on to chemical structural complexity, you have to know that a certain amount of chemical structural complexity is required for molecules to become immunogenic. And complex proteins with numerous and diverse epitopes are more likely to induce immune response than simple peptides containing only single or few epitopes. For example, amino acid homopolymers are less immunogenic compared to heteropolymers, which have 2 to 3 different amino acids. Moving on to epitopes, or what we also refer to as antigenic determinants. So these are small chemical groups on the antigen molecule that evoke the reaction with the antibodies. And they are the smallest parts of an antigen that can be seen by somatically generated B and T receptors. So an antigen may have one or more epitope, and you need to know that most antigens have multiple epitopes. and they are referred to as multivalent. So different epitopes on the same antigen may be recognized by different lymphocytes, each with a unique set of receptors. Regarding conformation and accessibility, you need to know that epitopes must be seen and be accessible to the immune system in order to induce an immune reaction. And for chemical properties, a protein immunogen must be enzymatically cleaved by the phagocytes for it to induce immunogenicity. For example, L-amino acid containing polypeptides are usually good immunogens, whereas D-amino acid containing polypeptides are poorly immunogenic. Regarding genetic constitution of the host, the genetic constitution of the host determines whether a molecule is immunogenic or not. For example, different strains of the same species of animal may show different response to the same antigen. Moving on to dosage, route, and timing of antigen administration, you need to know that immunogenicity is also affected by these factors, in the sense that very low doses of antigen do not stimulate immune response because either too few lymphocytes are contacted or because of a non-responsive state is elicited. On the other hand, an extremely high dose also fails to elicit tolerance. This is why repeated administration of antigens, that is the booster doses, may be required to enhance immune response of the host to certain antigens. And this is particularly important in the case of vaccines, where a prerequisite immune level needs to be attained, and the booster doses of vaccines are given to ensure good protective levels of the antibodies. Moving on to adjuvants. So adjuvants enhance the immune response to an immunogen, and however, they do not covalently bond to the immunogen. So they act in various ways. Number one, they can cause slow release of antigens. They also enhance uptake of immunogen by antigen-presenting cells. They induce co-stimulatory molecules, and they can also stimulate toll-like receptors. Moving on to biological systems, so some substances are immunogenic in one individual but not in others. So we have those ones that are responders to certain substances, and others are non-responders. And this is due to the fact that individuals may lack or have altered genes that code for the receptors for antigen on B and T lymphocytes, or they may not have the appropriate genes needed for the antigen-presenting cells to present the antigen to helper T cells. So the next thing we will discuss is antigenic specificity, and specificity basically refers and also depends on antigenic determinants or the epitopes. So it refers to the mechanism in which an antigen binds with the antibody to elicit an immune response. And binding takes place at the epitope. So an epitope is the immunologically active region of an immunogen that binds to the antigen-specific membrane receptors on the lymphocytes or secreted antibodies. So we have two types of epitopes, the B-cell epitopes and the T-cell epitopes. So B-cell epitopes are antigenic determinants recognized by the B cells, and they can combine with its receptors only if the antigen molecule is in its native state. And B-cell epitope is about 6 to 7 amino acid long, and they are hydrophilic. Moving on to T-cell epitopes. So T cells recognize amino acids in proteins but do not recognize polysaccharides or nucleic acid antigens. For major histocompatibility molecules and peptides, they are recognized by T cells. Therefore, T cell response is MHC restricted. And T cell epitopes are about 8 to 15 amino acid long. Lastly, we are going to discuss how Hapten carrier conjugate induces an immune response. So first question, Hapten carrier protein complex will interact with immunoglobulin M receptor on the surface of the B lymphocyte. And as a result of that, the hapten carrier protein complex will be internalized, and then the peptide from the carrier protein will be presented in association with class 2 MHC protein to T helper cell. And this will activate the T helper cell, which then produces different types of interleukins, for example, interleukin 4 and interleukin 5. So B lymphocytes will also be stimulated to become the plasma cells, and the plasma cells produces antibodies that will be specific for the hapten. So this concludes our second presentation. I hope someone finds this helpful. In case you like my presentations, please share, comment, and also subscribe on my channel for more presentations. Until next time, keep safe. Thank you.